Good morning, everyone. I am Dr. Rna Banerjee, working as an assistant professor at the Center for Health Science and Technology, JIS Institute of Advanced Studies and Research, Kolkata. JIS Institute of Advanced Studies and Research, Kolkata is an unique postgraduate research institute under the aegis of JIS University. JISISR started its journey 1 year back under the leadership of Padma Shri Professor Ajay Kumar Roy. We are located at Sector 5, Salt Lake. I welcome you all on behalf of JISISR. We offer industry-oriented postgraduate and translational research program in some of the most relevant areas of science and technology. Currently, we are having 3 centers: Center for Data Science, Center for Health Science and Technology, and Center for Interdisciplinary Sciences. Today's webinar has been conducted by the Center for Health Science and Technology. In short, we call it JIS. In the Center for Health Science and Technology, we offer 2 master's programs, MTech in Bioinformatics and MSc in Medical Biotechnology and Bioinformatics. These programs are carefully chosen in response to ever-increasing call of technological expertise in both academic and industry sectors of health sciences. And today, qualified bioinformatics professionals are in high demand of top salary jobs worldwide. Surprisingly, while such a field is of so demand and such programs are being offered everywhere across the world, we are the only institute in Kolkata offering postgraduate courses in bioinformatics and medical biotechnology. We also have PhD and integrated master's PhD program in bioinformatics, medical microbiology, cancer biology, and biomedical engineering. And here lies our uniqueness. We represent an unique research-based education system with special trust in health and medicine, keeping in mind industry-oriented real-life problems. We have advanced molecular genomics, biotechnology, and bioinformatics labs, internationally experienced faculty team, prestigious high-budget ongoing research grants, extensive collaborative research with multiple Indian and US companies, and with renowned labs of several US and German universities. Several ongoing research projects funded by DBT, ICMR, DBT Wellcome Trust, DBT Ramalingashami Fellowship are available in our institute. Let's have a quick look at our research focuses. We are interested in research related to bacterial virulence and antibiotic resistance. We are interested in the analysis of human microbiome and metagenomics. We are also focused towards molecular modeling and drug designing, medical microbiology, medical imaging, and machine learning. We are having high-end laboratory facilities with avant-garde equipments like Illumina MySeq sequencer machine. We also offer computational services related to huge data analysis. Illumina MySeq sequencer offers DNA sequencing like small whole genome sequencing, cheap sequencing, amplicon-based targeted gene sequencing, targeted gene expression profiling, 16S metagenomic sequencing, etc. This is our faculty profile. Our faculties are having patents and publication in premier journals like Nature Group and Proceedings of National Academy of Sciences and so on. Visit our website for more details about our faculties. The foremost objectives of our MSc and MTech programs is to offer the students a solid foundation on all different aspects of computational biology and bioinformatics applications throughout the 4 semesters. Admission is going on in our institute. If you feel interested, feel free to call us for any query in the phone number provided here, or drop a mail at biosimp@jisisr.org. Also, you can visit our website for online application, and you can also visit our Facebook page. Now, time for today's lecture. I feel privileged to introduce my friend, Dr. Srimari Mukherjee. Dr. Srimari Mukherjee is a postdoctoral scientist working at the Tufts University School of Medicine, Boston, USA. She did her PhD from University of Calcutta, Department of Biophysics, Molecular Biology, and Bioinformatics. All through her career, Srimari has been working on the mechanism of gene regulation in immune cells. She was the only researcher in her PhD lab to delineate the transcriptional regulation of multifunctional cytokines. She also worked on post-transcriptional regulation and effect of miRNA in metastasis. She has an impressive number of publications in internationally reputed journals. For the last 2 years, she has been associated with Moore Lab, known for being one of the pioneer groups in establishing the roles of important components of mRNA polyadenylation machinery in yeast. After joining Mood Lab, Srimari has been established the human immune cell system independently. She is currently interested to decipher the role of alternative polyadenylation in this system. Her outstanding research in this field has earned her the Napoli Zucker Award for Women Scholars 2020, which is a year-long grant to further her work. Let's listen to Srimari, and please don't forget to write your queries in our question and answer section. Srimari will be happy to answer you. Over to our speaker, Srimari.
Thank you, Dr. Banerjee, for introducing me with such kind words. Am I audible?
Yes, you are audible.
Okay, good morning, everyone. This is Srimari Mukherjee, live from Boston, United States. I would like to extend my warmest thanks to this institute for reaching out to me for this opportunity to talk about my research at the webinar 3 organized by JIS Institute of Advanced Studies and Research. This is indeed a very enriching initiative that they have taken to foster scientific thinking among students in this phase of pandemic and also leading to exchange of scientific ideas among us. So I will share my screen and start talking about my research. Okay. Yeah, do you see my screen? Is it?
Yes, your screen is visible.
Okay. Yeah, so the topic of my research is the role of alternative polyadenylation in macrophage differentiation. And this is what I'm doing for my postdoctoral studies in the Moore Lab, as she said. So I will try to keep things pretty basic, keeping in mind that there are students who are participating in this webinar and I don't want to alienate them. So feel free to shoot me questions later on or between the talk. I will be happy to answer them for the ones who are more interested in these topics. So before I go into my system and why we are doing this, I will introduce some basic things like the immune cell classification, because we are working with the cells and the monocytic and macrophage cells. So I would like to introduce what and how they're classified. First of all, starting off with the hematopoietic stem cell, they generally differentiate, they can differentiate into generally common myeloid progenitors or lymphoid progenitors, depending on how the stimulus is. And then the common myeloid progenitor can get differentiated into megakaryocytes and later on into thrombocytes, which we commonly known as platelets, erythrocytes or RBCs, mast cells, myeloblasts, and these myeloblast progenitor cells can get differentiated into basophils, neutrophils, eosinophils, and monocytes. Now, these monocytes can get differentiated into macrophages and dendritic cells, and there are different kinds of macrophages which I'll come into later. And this differentiation happens according to what the stimulus is and how the cell condition is, or what the inflammatory stage of the cell is. On the other hand, the common lymphoid progenitor can get differentiated into the natural killer cells and small lymphocytes, which originates to T lymphocytes and B lymphocytes. And these B lymphocytes are differentiated into more activated plasma cells, which are responsible for secreting a lot of antibodies and helping us in combating a lot of infections. So from all these immune cells, generally the macrophages, neutrophils, and the dendritic cells are the phagocytes. That is, they are the members which reach the inflammation site irrespective of any direction, irrespective of any memory, or irrespective of any stimulus. The mast cells, the eosinophils, the basophils, and the natural killer cells, these are components of the innate immunity, which are also the first line of defense, whereas the T lymphocytes and the B lymphocytes are generally the second line of defense, and they constitute the adaptive immunity, which has got memory, and it prepares the body for more complex immune functions, and also it remembers the pathogen, and it can, get back to action when the pathogen attack is again happening. So which we are interested in is the monocyte to macrophage differentiation pathway. Firstly, because these monocyte macrophage differentiation is the first line of defense, and they are the first activators when any kind of inflammation happens or any pathogen attack happens. So we wanted to find a therapeutic angle to differentiation of these lineage. So why monocyte and macrophage differentiation, and how can it be used as a therapy? That is what we are concerned with. First of all, as I've said, that like the common myeloid progenitor getting differentiated into myeloblasts and monocytes and then the macrophages. These macrophages are generally at the M0 stage right here, the naive macrophages, which are not polarized into anything. Depending on the stimulus, they can get differentiated into M1s and M2s. These M1s are the pro-inflammatory macrophages, and M2s are the anti-inflammatory ones. But normally, these differentiation pathway occurs in our bodies, and that is the normal course of differentiation. But in any case, if the differentiation is inhibited at any of these stages, it can cause accumulation of a lot of cells just wherever the inhibition happens, and then it can cause a number of inflammatory response and accumulation of a lot of cells and division, more and more cell division, which can lead to a disease like cancer. And acute myeloid leukemia is what we call the cancer of this kind of lineage. So any therapeutic angle taken with using all these cell lines will always be directed towards eradicating leukemia. And because leukemia or AML, as I've said, is diagnosed in about 120,000 in cases worldwide in 2017, it's really important that we find a good way to get rid of this. So as a general therapy, we generally depend on traditional therapy like surgery or chemotherapy or radiotherapy to get rid of the cancer. In other words, kill the cancer cells and get rid of the tumor. But this can lead to a lot of side effects, like it can also have an effect on the normal cells, and the traditional therapy is not always, it doesn't always eradicate all the cancer cells. So there are many alternate therapies, and one of them is the differentiation therapy, which is proven to basically be very effective nowadays. And ATRA or arsenate or 13-cis-retinoic acid, as I've mentioned here, has been used as agents which can differentiate the cancer cells. So what it does is basically, wherever there is any block, and whenever the cells are not differentiating and they're dividing in numbers, the differentiation agents basically work on these cells, and it kind of forces them towards differentiation by deleting the block here. So what they do is, like substances like formal diesters, etc., or they have worked in cell lines like HL60 or K562. These are all AML cell lines. And this kind of approach is basically used to treat cancers by overcoming the block differentiation, as I've said. So now the blocked differentiation is, if they overcome that, they will again differentiate into the normal course, and then it will follow the normal course. So now whenever differentiation happens, or whenever there is any differentiation event in any cells, there are a lot of gene regulation mechanisms taking place. And of course, alternative polyadenylation is one of them. I will get into what it is and how it works. And it can be globally regulated in response to changes in cellular proliferation and differentiation. So we were very interested with this system, and also, with this system. Firstly, as I've said, I will just take a few minutes to move over why this system again, just so that you can understand the importance, because it has significantly implicated as the first line of defense, as I've said. Monocytes have characteristic immune effector functions, having chemokine and adhesion receptors that mediate migration from blood to tissues during infection. They can differentiate into inflammatory dendritic cells or macrophages depending on the inflammatory situation. And monocyte to macrophage differentiation is significantly important in preventing neoplasm. Because as I've said, this is considered as a differentiation disorder or de-differentiation disease. So this system became our prime concern, and we were working, I came here and started establishing this system. So I will just go into how we measure monocyte macrophage differentiation and what surface markers are specific for these. So each cell, as I've shown before in the immune cell classification, each cell have their own characteristics with their own specific markers or cell surface receptors by which we can know that this is monocyte and that is macrophage, or that is M0 or that is M1. So the typical characteristics of each of these cell surface markers are really different, and that is how we detect them using either Western blot to monitor the cell surface proteins, or FACS, or other methods by which you can monitor the cell surface markers primarily. There are other ways as well, but this is the primary way. So what we generally have planned or devised, or what people use, are we've used the monocytic cell lines, U937s and THP1s for our study. We've also worked with mouse primary monocytes and human primary monocytes. I will not complicate things by including those things here, but I'll just stick to the cell lines. We have treated them with a Forbol ester PMA, which results in the differentiation of these monocytes into macrophages, essentially naive macrophages, that is the M0s. So if somebody wants to polarize, okay, before that, the macrophages can be characterized by a decrease in CD14 receptor molecule, which is a monocyte marker. So as the cells differentiate, the CD14 receptors will show up less, and the CD38 marker, which will show up more when the macrophage differentiation happens. And they can get differentiated into M1s, which are the classical ones, or M2s, that is the tumor-associated ones, depending on what kind of stimulus we are using. We haven't gone into M1s or M2s as yet. We will just stick to monocyte, the macrophage M0s for now. But we have plans to consider these polarized macrophages as well later on. Firstly, because M1s are totally different from M2s, M2 macrophages. And these are the agents which can differentially polarize these macrophages to either M1s or M2s. So M1s are the classical macrophages, which are mostly pro-inflammatory, microbicidal, and anti-tumoral. That is important because M1s are the good macrophages, which are helpful in eradicating the tumor, or they are pro-inflammatory and they are resistant to tumor formation. Whereas macrophage M2s are tumor-associated macrophages, that is, they work for the tumor, they are anti-inflammatory, they prevent any inflammatory response from the body, from the host, they are responsible for proliferation, and they are pro-tumoral. So the agent that we are using, PMA, or the differentiation agents that have been used to treat AML or have been in trials, they always want these macrophages to differentiate into M1s, which are the classical ones, so that they have this anti-tumoral activity, and so that it can be really deleterious to the tumoral cells. So classical macrophages can be categorized by these surface markers, and M2s have their own surface markers. So this is how we define them, and we've been checking these markers to always monitor whether they're more macrophage or more monocytic. Now, the systems that we've been using, I've said, these are the cell lines, and this is how we differentiate them. So whenever we differentiate, that is, we treat them with PMA or the Forbol ester that I've been talking about, what they do is basically by 6 hours of PMA treatment, these cells get really adherent to the plate. That is, they attach to the plate, to the surface where they're being cultured. And by the end of a day, they completely attach to the plates, and they have elongated structures now. They perfectly look like macrophages, and this is a close-up of how they look like. They really stick to the surface. They have pseudopods, and they have macrophage-like properties. And this happens with the cell lines. We've also established the mouse monocyte system. We've also work with them, and they also differentiate really well with macrophage stimulating factor. And the same case with human primary monocytes isolated from whole blood. So these are the various differentiation models which we've been using to delineate the mechanism of APA. And the evidences for differentiation, the main experiments that we do are generally measuring the cellular complexity or granularity. I have just shown you a sample figure so that it's understandable. So I've just included a U937 cells figure. So basically what we do is we fax analyze the cells without PMA treatment and with PMA treatment, that is before differentiation and after, and measure the side scattering and the forward scattering, which will measure the cell size, cell complexity, or cell granularity. The more the macrophage formation takes place, the cells become more heterogeneous and more complex and more granular. So you can see more scattering and formation of probably two populations, as if you could see here, which is some monocytes and some really differentiate into macrophages. So this is clear that the macrophage differentiates, and we can also measure how many by gating if you want. Another is, as I said, the surface markers are really different from cell to cell. So we measured the surface markers also, like decrease in CD14, increase in CD38. ICAM1 is another macrophage specific receptor which increases with differentiation. And another assay that we have included, or we do, is because I've said the cell attaches to the plate, or any macrophage has this property of getting attached, and they are not in suspension anymore. So what we do is we use the attachment assay. We do the attachment assay by trypan blue counts of cells which are suspended and which are attached. So we basically treat the cells with PMA, collect the suspended cells after a day, and then collect the attached cells by trypsinizing them or by detaching them from the surface, and then take account of that as well. So we do this in triplicates, and this is how we represent the results by a bar graph where the green ones show how many are attached and the pink ones show how many are suspended. So you can, as you can see from the 6-hour and 24-hour treatment of PMA, almost 70% of the cells have got attached by 6-hour, and almost 90% have got attached with 24 hours of treatment. So this is the normal course of U937 differentiation, and this is how we measure it. Also, along with attachment, there comes the less proliferation rate. So as I've said, when the cells proliferate a lot, they don't differentiate, and when the cells differentiate, they don't proliferate. So by measuring the proliferation rate using a biochemical assay, we could show that with PMA, without PMA treatment, the cells double normally, but with PMA treatment, gradually, there is no doubling, and the proliferation rate is really low. Okay, so differentiation and gene regulatory network, they have a unique connection because differentiation is a huge process which takes place in the monocytes, and it changes the cell shape, the cell property, the cell biochemical properties, and all the gene expression properties. So basically, when we treat these monocytes with any differentiation stimuli as such, it can have an effect on the change in expression of core proteins of gene regulation, which can be anything, starting from RNA polymerase to transcription factors to mRNA binding proteins to splicing factors, anything. And also they can modulate the cellular pathways like PK pathways, MAP kinase pathways, and all these pathways that we know, which can have an effect on the transcription factors and the protein markers like the cell surface markers, the integrins, glycolytic pathway, ferritin, ROS production. And finally, the macrophages, and they secrete specific cytokines and chemokines. All these factors and all these chemokines and the proteins which are expressed by macrophages, they can all be under the control of gene expression. And at any level, like transcriptional, post-transcriptional, translational, any kind of control of gene expression can happen. And this is a fairly complicated process which has not been elucidated in full. till now, and we are still just doing a part of it. So going into the mechanisms of gene regulation, what happens basically, it's almost known by all, but just to give you a basic idea for those who still are students, is first of all, it starts with a chromatin decompaction, and which is obviously the epigenetic regulation, followed by the transcription regulation at the initiation, elongation, and termination level. So the transcriptional regulation is an important feature of any process which determines the amount of RNA produced. I have been working on transcriptional regulation of a cytokine in the same system that I'm working with now. So I have been working in U937s and monocytes and macrophages for a long time, since my PhD. So I have worked on the transcription of a cytokine during differentiation, and that was an interesting piece of study. After that, the transcript cleavage happens and it moves on to the RNA processing pathways like capping, 3' polyadenylation, which is what I'm concentrated right now, splicing, mRNA packaging, mRNA export, and of course, mRNA stability. Stability is something which I was associated with when I was doing my PhD. There were some things that I did with mRNA stability. So basically, the concept was the more the mRNA is stable, the more protein it produces, because it is associated with more stabilizing proteins. So it stays in the cell for more longer times, and so more protein is produced. So that is, and the RNA which are degraded very early, they cannot reach the polysomes, and so protein production is really less for them. So that is how it regulates the protein expression. And finally, the protein folding and translation and post-transcriptional modification. So what we are interested right now is with the RNA processing pathways, specifically the 3' polyadenylation. The cleavage and polyadenylation that happens with the 3' end of all eukaryotic mRNAs and link RNAs transcribed by RNA polymerase II is really an important process. It is tightly coupled with transcription termination because it happens at the same time or in similar times. The poly A signal is defined by the surrounding sequence motifs. We've been reading about poly A signals and how it works for a long time, but to just give you a brief overview of what exactly happens is, if this is a poly A signal that we see here, AAUUAA or AUUAA, as we've read before, they are flanked by an upstream element and a downstream element. This upstream element is generally a GU-rich element, and the downstream element is a GUU or U-rich element. These are bound by specific complex proteins, which I'll come to later to their exact descriptions and what they do. So basically, the CF2M complex occupies the upstream element, followed by the CPSF complex, which is the main complex, and one of these, CPSF73, is responsible for the cleavage here. They bind to the poly A signal, and the downstream element is bound by the CSTF complex, and there are other core proteins as well, which are not included in this figure. We'll come to that. The PAP or the poly A polymerase is then responsible after the cleavage signal is activated and the cleavage is done. This is responsible for coming here and constructing the poly A tail. And then the PABPN proteins are also necessary to make this more stable. And then the poly A tail, once it is formed, the mRNA is basically transported for translation. So what if there are more than one poly A signal in an mRNA? That can lead to isoforms of polyadenylation, which we call alternate poly A isoforms or APA isoforms. So I will be using APA. The term APA means alternative polyadenylation. And this is very tissue specific. It has got dynamic role in cell proliferation and differentiation. And the core CPA proteins, CPA stands for cleavage and polyadenylation. These expression level changes are found to be potent in carrying out their effects on cell proliferation and differentiation. That was found out long back. So why is it important to study? First of all, the choice of a transcripts Cleavage polyadenylation site occurs in the nucleus, and it is an important process, regulated process, as I have said. APA can lead to changes in transcript stability, localization, splicing pattern, translation, and the protein product generated from the gene. At least 70% of the mammalian transcripts are alternatively polyadenylated. The majority of alternate transcripts is due to APA and not alternative splicing. So you can imagine how important it is after transcription. And very few mechanisms controlling APA have been elucidated despite its predominant role in gene expression in human health. And to our knowledge, there is no report of mechanistic role of APA in monocyte macrophage differentiation till date. So it was a very interesting piece of study that we started with. Let me now continue into what the consequence of APA can be in cells. So imagine this is an mRNA, typical with a cap and a 3' end, and it has got 3 exons and a 3' UTR and like 2 introns, as you can see. And imagine that there are 3 poly A sites for this mRNA instead of 1. One is inside the intron, one is in the proximal end of the 3' UTR, and another is at the distal end of the 3' UTR. So for example, if the cell chooses this PA site, then it will cleave here, and this will produce a non-functional protein or a protein with an altered function, which will not have the exon 3. So this can have a total group of consequences, like with either non-functional protein or altered function. I will come to a classical example so that you can understand that. Next, if the mRNA chooses this PA site, that is the proximal one, then it will be a shorter 3' UTR transcript, but protein production will be there. And if the cell chooses this PA site, then there will be production of the long mRNA, long 3' UTR mRNA. And then this long UTR can have regulatory binding sites more than this shorter transcript, and more so that this can be functional with respect to the binding of either RNA binding proteins or microRNAs or link RNAs. And it can lead to mRNA stability, mRNA translation, mRNA nuclear export, or localization and protein localization, as I've said. So it is believed that the longer the UTR, it is more prone to regulation. So normally it is considered that if the proximal PA site is chosen, then the cells will have an increased CPA protein amount, that is the core protein complex expression will increase. And also the target expression will also increase. Like if this is a shorter isoform, then it will produce more protein just because it doesn't have all these regulatory sites where microRNAs and destabilizing proteins can bind and can shorten its half-life. But if the distal PA site is chosen, it is generally believed that there is decrease in CPA proteins and also decrease in target expression because this is subject to a lot of regulation. But honestly, this general theory is not true for all the systems, and we ourselves observed something different and interesting. So I will come to that. Let me just introduce to you why and how APA is important. We all know about the transmembrane IgM and a secretory version of the IgM. Now, interestingly, the transmembrane IgM and the secretory version of IgM, they originate by the usage of differential PA sites. So for example, this transmembrane IgM has got, the RNA has got 2 PA sites. One is the proximal one, another is the distal one, if it could be seen clearly. So when it chooses the distal PA site, it has an inclusion of the C terminus for the transmembrane IgM, as you can see here. And this is a bound IgM that's produced if the cell chooses this PA site. Whereas if the cell chooses the proximal PA site, then it doesn't have the C terminus for the transmembrane IgM at all, but it's just somewhat short, and it produces this, the secretory IgM. So this is one of the examples where APA really plays an important role in deciding what kind of protein it will produce or what kind of protein structure or functional consequence of what protein is being used. So this is how APA is important, and this is just one example, and this has been found long back. So since then, studies with APA has become really important. So our strategy to monitor APA was, first of all, qPCR based method. I did some rough analysis on what happens on monocyte macrophage differentiation, whether there are any polyaside switches, or if there are changes from shorter to longer or longer to shorter, and what happens to some target genes. So let me introduce the strategy that we use. This is a well-accepted strategy where we devise two sets of primers, one at the coding region, which will measure the total mRNA, that is long, short, everything, all size. And another primer pair is designed between the PAS1 and PAS2. PAS is the polyA signal 1 and 2. So this primer pair would only amplify the longest primer, the longest transcript. So we get the total and the longer transcript by choosing two sets of primers here and in the two different positions. And then we measure the ratio of the long by total transcript. So if the long by total transcript ratio is less than 1, then we can imagine that the PAS1 was chosen, that is more of the shorter transcript was produced. But if the ratio is more than 1, then we can imagine that the PAS2 was chosen overall, and more of the longer transcript was produced. So this is very quantitative, and it gives us an initial idea of how the APA status of the cells are. So we got some idea about what happens in monocyte macrophage differentiation, and I've seen with some of the target proteins, which are really important, like some transcription factors, JISIASRs, and some cytokines. They showed some kind of shortening, like I found the ratio to be less than 1 for most of them. So then by just by eye, we were choosing some factors because we knew that were important for macrophage function, but we wanted to have a global landscape of the APA changes by a sequencing method, so that we can know the global scenario of monocyte macrophage differentiation, which has not been delineated as yet. So we used a version of RNA-seq, which is the poly-A-click-seq method, also called the PAC-seq method. It is very sensitive and specific enrichment for polyacide junctions occur in this RNA-seq method, and there is no complex sample preparation, fragmentation, and purification involved. So this is very specific to amplify the last exon and the junction of the PA site with that. So I will come to how they designed the experiment and why PAC-seq is really specific. But as you can see from this graph, a comparison of normal RNA-seq by PAC-seq, the terminal exon is highly enriched in PAC-seq. So what we want is, we want to know the exact PA site choice for each of the condition. Like if we are choosing monocyte and macrophage, the two conditions, we want to know what PA sites are chosen during the monocyte form and which PA sites are chosen for the macrophage form, so that we can understand whether there are shortening events or lengthening events when the differentiation happens, and why does it happen. So we chose this method. I will just go into the steps so that you can understand what they use. So basically, these are the steps of PAC-seq library preparation, where they do a RT-PCR with azido VTPs. Azido as in, it has a chemical crosslink at the 5' end. And they use all the azido VTPs as a spike into the normal RNTPs. But they don't use azido TTPs because they don't want any termination at the poly-A tail. There is P7 primer, which is the Illumina specific primer attached at the 5' end with a 21 T sequences. This is an unanchored poly-T primer, followed by a combination of 4 nucleotides, and then an N3 here. This N3, or this chemical substrate at the 3' end, is prone to chemical cross-linking, which I'll talk about. So if a transcript A is the long transcript and transcript B is the shorter transcript, so this is how they will anneal. And the poly-T primer will anneal at the poly-A tail, and then it will anneal to the junction of the poly-A tail and the exon for both cases. And then the chemical reaction of click-sick, as they call it, a click-sick is basically a chemical reaction where the P5 primer with a chemical substitution is click substituted. It uses a chemical compound CuAC, which is a complex reaction, so that it forms a chemical bond with a 3' end wherever the termination has happened. And then they perform PCR, and this is how they look like. The P5 and the P7 at both ends with the fragment in between. And they gel extract the final library and cut the 200 to 400 nucleotide fragments only. And the final library topology looks like P5 and then followed by a part of the 3' UTR, the junction of the 3' UTR and poly-A tail, and 20 or 21 nucleotides of the poly-A tail. So we definitely get to know the junction where the PSI choice is made, or where the cell has chosen to be the termination site, as well as the length of the poly-A tail is also sometimes important for the stability or for a lot of research. A lot of research is also going on with the length of the poly-A tail. We are not yet thinking about that thing, but we wanted to know the last exon and wherever the poly-A sequence is added to the transcript. Overall, we can know that whether the shorter isoform or the longer isoform is being expressed at each defined condition. The processing pipeline, we have used the NextSeq 550 obtaining 1:150 base pair. We had a 130 million paired end reads, and it's pretty deep because we wanted to have a deep sequencing so that we can monitor all these changes. And the processing pipeline looks basic with raw reads in FASTP or FastQC files, and then trimming them and then star aligning them to the wet graph files, then sorting the reads, and they run it through a DSeq2 pipeline, whereby we have the normalization, we have the counts, we have the principal coordinate and differential expression of each of these genes, and whether they were shortened or lengthened according to where the PSI choice was made. And we can also visualize them using the bed graph files and uploading them to the UCSC genome browser, which I will show you. So we have a list of genes. We have an Excel sheet with, as a data, we have an Excel sheet with all this information. From there, the results that I've got with the high throughput pack sequencing for the data sets that we've been using, the monocytes treated with differentiating agent. So for the 0 to 6 hour, we've done a 0 versus 6 and 24. So the course of differentiation we've checked. So for the 6 hours, you can see around 12,000 genes were detected, and 12,000 for both the 6 and 24 hours. In the 6 hour point, you could see that there are 139 shortening events and there are 113 lengthening events. So a bit more of a shortening event, but there was both. And in case of 24 hours, there are much more shortening events and much more lengthening events than the 6 hour, but still they are comparable. 376 and 306 are very close to each other. But what I mean to say is the shortened and the lengthened transcripts are very well distributed, and there are equal numbers of these the mRNAs undergoing shortening or lengthening. So when we wanted to check whether the shortening resulted in an upregulation of the mRNA or downregulation, or vice versa with the lengthening, because I have already said that shortening of a transcript could mean increased expression. So we wanted to just see from our data sets how this looked like. To our surprise, we could see that there are shortened upregulated ones as well as lengthened downregulated ones, but there were also lengthened UTRs which were upregulated and shortened UTRs which were downregulated, which is contrary to the normal belief that shortened ones will always get stabilized or lengthened ones will always get destabilized. So this is the same case with 24 hours as well. So we can take home from this data that it's not always very straightforward that all shortened ones are stabilized and all lengthened ones are destabilized. So there could be mechanisms for these lengthened ones which are upregulated and shortened ones which are downregulated, and we will move into that later on. But yeah, I showed this data because this was very interesting, and this was on contrary to the normal belief that it can not go both ways. But there are some recent reports which has come up, which has also showed that they have found equal lengthening upregulated events along with lengthen downregulated and vice versa. So more observations are coming up when more deep sequencing methods are being used. And as I said, we could also visualize genes which were shortened or lengthened through bed graph files. This is how the bed graph files look like. So basically, the height of the peak and the height of the peak or the area under the peak basically represents how many events are taking place, or how many were detected. What was the signal like? So I will show you just like 4 genes which were very important to macrophage biology, and they were undergoing shortening and lengthening, or right, like polyacide switches on differentiation, which was really related to the macrophage system when we were interested. So these are some of the reviews that I also, or the papers that I attached, which says why they're important to macrophages. First of all, from the shortened groups, those that showed shortening, that is choosing of the proximal PA site. One is the O-linked N-acetyl glucosamine. You can see here from the 0 hour and the 24-hour point, if this is the proximal PA site and this is the distal PA site, as the annotation says. The peak here shows a full peak, and at 0 hours it doesn't choose, it almost chooses a very little amount of the proximal PA site, whereas on differentiation it considerably chooses a lot of the proximal PA sites now, which clearly mentions that on an average it has got shortened. Same case with the serpin B1, which is also very important in regulating inflammatory responses, and that is why related to the macrophage biology. It also shows that the distal poly-A signal, it decreases to a great amount with differentiation, which means the shortening has taken place. Some of the lengthened groups, let's have a look to arrestin beta 1, which is important for the complement expression in macrophages, as this report says, and it has got many other functions as well. As you can see here from the 0 hour point, the control ones this does show a long peak here, whereas this PA site is not very well chosen when they are differentiated. So it shows lengthening. And the same case with TGF-beta activated kinase 1, which is also a very functionally important protein, and it also shows considerable lengthening, that is increase in this distal PA event on differentiation. So these are just some of the targets that we found from the data set, and this data set is very huge. We are still working on this, and hopefully we'll get some targets which are more related to macrophage biology, and we can work on them later on. So coming to the core components, because we saw all these shortening and lengthening events, we were interested with seeing what roles does the core components of the mRNA polyadenylation have. So as I said before, the upstream element is occupied by the CF1 complex, consisting of 68 kilodalton and a 25 kilodalton subunit. These are responsible for binding the RNA with preference for the polyadenylation precursor. And the poly-A signal is occupied by the CPSF complex, which is the main complex. The CPSF73 is responsible for the cleavage. 160, 100, and FIP1, these are also, their functions are known. They are responsible for binding to the poly-A signal, interacting with the poly-A polymerase, and also interacting with the other proteins on the upstream and the downstream elements. The CSTF complex, which is bound to the downstream element, this breaches this, both these complexes and also binds to this U-rich sequence. The exact function of all these proteins are not yet elucidated, but it's still going on. And another complex, CF2M, has been recently found out, which consists of the PCF11 and CLIP1. These are also important regulatory factors, and they have important consequences on the lengthening and shortening or choosing of the PA sites. So various studies are still going on. And also the poly-A polymerase, it catalyzes the poly-A synthesis. This is also part of the complex. And there are other components like symplecine and all these proteins. They have their different functions, which all of these functions are not yet known. My PI had been delineating the mechanisms of the yeast polyadenylation system. And now we are working on the mammalian, the human system now, and trying to elucidate their roles. So in trying to elucidate the roles of these proteins, we chose the ones which were directly bound to the RNA, like CF125, FIP1, and 64, CSTF64. These were the targets that we chose primarily to see whether they have an effect on the choice of the poly-A sites. So we could see with the global landscape that there are some switches and some changes of mRNA choosing one PA site over the other on differentiation. So there must be an activity of these complex proteins, and by modifying any of these complex proteins, we wanted to see whether they have an effect on cellular differentiation or not. So I'll just show you one piece of data, just not getting into complicated results. So what we did was we knocked down the core factor CSTF64, this one, by siRNA. I'll show you the siRNA data and I'll tell you what we did later on. So the CSTF64 knockdown resulted in decreased expression of CPSF1, PCF11 to an extent. CF125 was not very responsive to it, but there has been a decrease in some of these factors, which is common because they kind of act in concert, and obviously somebody's function will be dependent on somebody else's expression. Now interestingly, we checked the levels of the macrophage surface marker on knockdown of this core protein, and we saw that there was a decrease in CD38 levels. That is, the knockdown of CSTF64 had an effect on the macrophage property. It decreased the surface marker expression. Also, when we did the attachment assay, it was just a rough experiment just to know whether this works. When we did the attachment assay, it definitely showed up a significant decrease in attachment when we transformed them or transfected them with siRNA against CSTF64, which was really promising. So what we did was, I will not go into the detail of what we did, but we established a stable inducible CSTF64 knockdown cell line just to make it more direct and concise. And inducible, why? Because we didn't want the cells to always knock down CSTF64 because it is one of the essential cleavage polyadenylation proteins. So for the inducible system, we chose a doxycycline inducible system with GFP tag, of course, and a puromycin and a Teton 3G system, and a smart vector universal scaffold, which will work on the principle that when you add doxycycline, the transactivator protein will only bind to the 3G promoter sequence, and which will activate the transcription so that this gets on and it will produce a lot of shRNA, whatever is cloned here. So we got this shRNA against CSTF64. We got 3 of them. One of them really worked well. And then we transfected them into HEC293 cells to produce the lentivirus. And then we transfected this, infected the E937 cells with these lentiviral vectors and selected them. And then did some assays to see what the effect of CSTF64 was. So with the control cells, the control shRNA transfected cells and the CSTF64 knockdown cells, what we did was we induced them with doxycycline so that they produce the shRNA, and then we measured the levels of macrophage markers by Western blot. We did AP analysis by qPCR method, like I said before. We did FACS analysis to measure cell morphology, and we did the attachment assay for macrophage functions also. And all of these indicate that a knockdown of CSTF64 results in a decrease of macrophage markers levels, and reversal of the shortening event that was previously happening. That is when the control cells were transfected, we could see shortening of some of the targets. And with CSTF64 knockdown, we could see the change of the PS site, a PS site switch from the proximal PS site to the distal PS site now. So CSTF64 is found to be really have important roles in choice of the PS sites during macrophage differentiation. And the FACS analysis and the macrophage properties were also to a great extent inhibited when CSTF64 knockdown was done. So basically, our overall findings till now was that we could establish the differentiation systems. We could see that there was an overall increased expression of cleavage polyadenylation proteins with macrophage differentiation, which is a novel finding. Unlike the established trends, mRNA is important for macrophage differentiation and function shortened during differentiation. That is what we found. Knockdown of CSTF64 slows differentiation and reverses the shortening of mRNA seen during differentiation. And the global landscape of genes show altered use of tandem sites in the 3' UTR or PA sites within introns, and how these changes were correlated with expression. That also we've seen with the PAC-seq. And we got an insight into the functional categories affected by different types of APA and identification of pathways important for macrophage function. And I just showed you 4 genes which were important for macrophage, but there were many pathways like cytoskeletal functions, cell division pathways, and a lot of cytokines and immunologic pathways coming up with analysis of the data set. So the questions that need to be answered still are, what happens when the other CP protein expressions are altered? Like we have worked with CSTF64, and now that we know that this has an effect on macrophage differentiation, we want to still show what happens with the other CP factors and delineate their mechanisms, and whether they act in concert with each other or individually, or whether they have a more enhanced effect. Do they also have similar effects or different effects? What the roles of CP proteins were in macrophage polarization to M1s and M2s. So by CP protein, I mean cleavage polyadenylation. And the roles of, so we haven't yet polarized the macrophages to M1s and M2s yet. We're just working with naive macrophages, the M0s. We want to know the roles of these proteins in the polarized M1s or M2 macrophages. By knowing that, we would be able to basically find target genes and target proteins which were important for choosing what pathway the macrophage will take, either M1 or M2. And by that, we can modulate the expression of surface markers or target proteins so that the M1s are more produced, so that we can have an antitumoral effect. We want to know the global landscape of PSI choice on the gene manipulation of CP factors. That is, when we knock it down, we will send the knockdown samples for PAC-seq again to see if there are global changes in the landscape as we see with the differentiated ones, and what the difference is with the CPA protein knockdown. What could be a possible target protein undergoing massive poly alterations which influence macrophage differentiation? So finding a possible target from these data set is really difficult, because these are it's a long set of data sets, but we are still trying to find out if there are specific targets like any transcription factor or a group of genes which gets modulated and which could be targets of the CPA proteins. That is, say for example, if a transcription factor undergoes APA and its expression changes a lot, and then this transcription factor can influence a lot of other genes and could be one of the possible targets for the therapeutic angle of treating AML. So this is what we are thinking about right now. And the roles of RNA binding proteins, splicing factors, and microRNAs in this whole process. So it's not only about the CPA proteins, it's also about the associated factors that are binding with it or interacting with it. We still don't know what they do and what roles they have. We just have some general idea and some initial data to start thinking about these questions. And what is the detailed molecular mechanism of the increased expression of long UTRs or decreased expression of short UTRs? As I said, that on the contrary to the common belief, we had lengthened ones which were still upregulated. So there should be some other mechanism by which they are getting stabilized, or the short UTRs, I mean the mRNAs containing short UTRs which are destabilized. So we want to know the mechanism of that population of mRNAs as well. So yeah, this is all. We've been lucky to get our first paper ready for to communicate right now, and these are some of the papers that I published before. And also as we work, we have been funded with the R01 NIH grant for 5 years for this project, and we are very happy to have started off with this project and made good success with it. And this has also made me one of the Natalie Zucker Women Scholar this year. I want to sincerely acknowledge the active support of my advisor, Professor Claire Moore, our collaborators, my colleagues, and all those who have been behind the experiments and everything that I do right now. And I would like to thank all of you for your time and your attention. Thank you so much. I'm open for questions now.
Thanks, Srimari. It was a wonderful talk. Thank you all for listening. Now we will have our questionnaires. The first one is from, okay, it was a very nice, just wait a minute. Yeah, it was a very nice talk from Muli Das. And also we have got, it was a very good presentation and excellent explained all the concepts from Mr. Ranojit Dash. And there is a question from Chandrama Mukherjee. Mm-hmm. Have you seen any kind of gene families to be enriched after your pack analysis?
Yes, we've seen a lot of gene families to be enriched. I didn't go into the detail, but honestly, the ones which are really important for our study were the proteins which, the pathways which were enriched were from the cytoskeletal aspect, because the cell differentiates. So this was expected that the cytoskeletal proteins will be changed, but they are considered as one of the important pathways to be enriched. Another one was with the cell division, which is also really important. ROS signaling, and yes, a lot of them. Chromatin modulation, if I remember correctly, and a lot more, yeah. They were enriched. And we are working on those things and trying to find out which one is the most important or the most significant. And mostly we are depending on the immunological aspect of it, so that we can have we can delineate the therapeutic angle of getting rid of AML by differentiation therapy. So yeah, the macrophage related properties are highly enriched in PAC-seq data. And
the next question is from an anonymous attendee. Can we artificially initiate monocyte macrophage differentiation to cure leukemia? That is, to improve immunological defense against leukemia.
Yes, that is what we are doing, because as I mentioned about the differentiation agents, those are all artificial agents. Like APTRA has been used a lot, and it has been used to it has undergone trials also, and it is used to initiate differentiation. And cis-retinoic acid as well. PMA is still, I think it has got some side effects still, because it is responsible for basically, activating the PKC and MAP kinase pathways. So that has got a huge effect, and we still don't know a lot of the consequence of these chemicals. But ACTRA, I know, has been implicated in artificially differentiating them. Yeah.
The next question is from Ranojit Dash. So you have talked about the role of alternative polyadenylation in macrophages. Has there been any evidence in any other immune cells?
Oh yes, the one that I talked about, the IgM secretory and the membrane-bound pathways, that happens in B cells. So that's one of the immune cells. APA changes have also been observed in T cells, and yeah, T cell reports are also there, and many other cells like fibroblasts and yeah, many other cells.
Okay, and Chandrama Mukherjee was who did the first question. I've given you thanks for answering. One thing to mention here, that the recorded version of this whole session will be available in our Facebook page as well as in our YouTube also. Those who have missed today can listen over there also. And Srimari, can you please stop sharing so that I can share the... So if you have any further query, you can call us in the number given, or you can drop a mail at biosim@JISIASR.org. Okay. Thank you all. Thank you, Srimari, for this nice talk.
Thank you so much. Thank you for having me here.
Thank you.